So I don't mind recording these lectures, but when nobody shows up, I feel a little less obligated to do it, even if one person does show up. But today I was actually thinking I was going to record it anyway, because I do kind of like talking about this stuff. And every time I talk about it, I find different things that I say sound better. So it's good practice if nothing else. But I do appreciate those of you who do get up and show up. So thank you, including my dog Cooper. He doesn't say much, but all right. What we're going to talk about is really no small task to me. It always kind of reminds me of walking into a movie in the last minute, sitting down and attempting to figure out what was happening in the previous however long the movie went on, right? Where you get in there, maybe it's a murder mystery or something like that. And at the end they reveal the murder, but you have no idea, no context. It's like my dad used to always get angry with me as a kid. I'd walk in halfway through in a TV show and start watching it, and I would say, "Why do you say that, Dad?" And he's like, "Oh, if you'd watch the show." So, this is kind of what the task that we face when we look at the history of life on Earth. We've been around for just a mere few last seconds of it, if you think about it. And you've seen that thing before where they take the clock and they met in 12. Was it 24 hours? Yeah, 24 hours. Where are we? And we show up. If you took all of history of life and put it on a clock, we show up in the last, I don't know, a couple of seconds before midnight, right? So trying to recreate or try to reconstruct what we think happened to life on Earth is really sort of a pretty monumental task. The fossil record helps us quite a bit, obviously. We've talked about that extensively. The fossil record gives us an idea of the critters that were around, and since we know quite a bit about that old niche thing, where critters fit in and what they do, we can sometimes make assumptions about where they lived. Make a simple analogy. Remember, Darwin found those marine fossils up on the top of a mountain. Okay, so not only did we get information about what types of organisms were around, but that told them that where they are now isn't where they were initially, right? So fossils give us clues of what the climate was like where the animals or the organisms survived. It also gives us clues about the dynamics of the Earth, the plate tectonics thing that wasn't really described for another 100 years or so. The real interesting part, well, no, they're all interesting, but the first very interesting part about the history of life on Earth is the first, right? When did we go from non-life to life? When did we go from rocks to things that really were alive? With all those definitions that we talked about in Bio 1, right? Like they're organized and you metabolize and all those things that I hope you remember. So the point is here, how did this happen? How did biogenesis happen? Fascinating topic. There are all kinds of things that you could talk about. There are all kinds of interesting theories. Some of them that years ago, even as I remember not too long ago, were seen as just crazy, just absolutely nuts, like the theories of I don't know, you could think of anything, UFOs or whatever. They're not seen as nuts so far, but you understand what I'm saying. But the theory I'm talking about, I'm going to type it out, is called panspermia. And the pan in science, pan means cosmopolitan. It means everywhere. So what panspermia says is that some planetary body, maybe a comet, an asteroid, or something like that, traveling around the universe, or planetary bodies, it would be many of them actually, might actually have the seeds to life. Maybe not, but maybe a certain key component of life. And that in some of these places around the universe, these comets or asteroids would make impact with this other planetary body, let's say the early Earth, and somehow seed life. Panspermia. Yeah, years ago seen as crazy, but now it gets a lot more evidence because, or gets a lot more credibility because we're actually finding on asteroids some of these things that we might call the seeds of life, complex organic molecules, even amino acids, those kinds of things. And there are critters or animals, folks, that we'll talk about later on called water bears. Look them up, tardigrades, tardigrades. Something like that in the spelling. Maybe it's an ES. Slow walkers, tardigrades, slow walkers. Really cool critters that can actually live in space. They take them out on the space shuttle and on the space lab and all that stuff, and they had a bunch of them in a box, and they put them on the outside of it, and they opened it up to space, opened it up to the radiation and the vacuum and the blah, blah, and no oxygen, and left them out there for 3 weeks, I think, or something like that. They brought them back in, and most of them were dead, right? Most of them were dead, as you would expect. Not all of them. Not all of them were dead. Some of them were still alive. Some of them were still alive. And the great question is, why would an organism have the genetic ability to survive this incredibly bizarre atmosphere or environment of space? Not even atmosphere, right? The vacuum, the radiation, and all the things that you know would kill any other living critter on Earth. Why would some of them have the genes to survive that? It's like a detective game. Maybe they got the genes from something else. Maybe it wasn't them necessarily, but maybe some primitive type of living or precursor of living had those genes, right? Because you see down in number 4, look what happens in number 4. These are steps, okay? We'll get to number 4 in the steps, but look at it right now. It says, toward the end of biogenesis, this theory, what we are seeing are molecules that make copies of themselves, self-replicating molecules, very unique, right? Very unique. Does carbon dioxide make copies of itself? You were in Bio 1, you remember this, right? If I have a bottle of carbon dioxide molecules, 1 million of them, and I come back in an hour, is there any reasonable reason for me to believe that there's going to be 2 million? What's that, Cooper, you say? Cooper says, no, of course not. But in certain molecules there were. You remember DNA and RNA? That's all DNA and RNA is all about. It's making copies. You make copies of the DNA, and then you make a type of copy when you make the RNA, and, you know, along comes life. Simple, right? All right, we'll get to that in just a second. But, you know, maybe those molecules, those self-replicating molecules, came on some of these asteroids, all right? So maybe what happened in steps 1, 2, and 3, maybe more specifically in 1, didn't happen. And yet once this self-replicating molecule was delivered to Earth by some planetary body, and the earlier solar system was a pinball gallery. That's what they say. There was stuff bouncing off and rocks, and we'll talk more about that later on. It wasn't a very nice place. Okay, let's go back to look at a theory of biogenesis. So let's assume maybe that panspermia didn't happen, something didn't deliver these special molecules. Or now the new theory is maybe it happened both ways. Maybe something delivered some of these self-replicating molecules, and maybe they formed. Let's go back and look at the very beginning, though. Let's follow what would have happened if, you know, they weren't brought from Earth, or in addition to being brought to Earth from something. Organic molecule formation. Remember, organic is important. There are very special types of molecules that are organic molecules. You spent a whole chapter on them in Bio 1. These are the molecules of life. They use carbon as their central element. Marie, do you remember why it was carbon that Mother Nature chose? Do you remember why it was that atom she chose, or became the atom of life, all living things having that atom in it? Do you remember what it was? Which element? Which element makes up an organic molecule? Four others. Yeah, but you get the right idea. Yeah, it was carbon. Remember, carbon has 4 valence electrons, 4 electrons in its outer shell, and it needs 8 to be full. But right, you understood the idea that what the atom was, more importantly, or that the question, was a multi-purpose atom, right? You could make lots of things with it. Remember in Bio 1, I showed you a picture of Tinker Toys. And I said, carbon reminds me of Tinker Toys because you can make lots of different things with it. So now, how does that all relate chemically? Remember that the whole idea of putting chemistry and biology, form and function together, that we keep emphasizing in this course. How does carbon's ability to be versatile, how does that link up with life? Life is really diverse, right? So if you have something that's versatile, or another word for that would be diverse, then you can make diverse things out of it. You see what I mean? If the little Tinker Toys only had one hole in them, if you remember that analogy, you can make so many things. But you put all them other holes in it, you make all kinds of things out of it. You can do the same thing with carbon. Okay, so we got to form these organic molecules. So let's go back to the way the early Earth was. Let's go back to the way the Earth was. Wayne's World. I wish I had some special effects to do that. 3.5 billion years ago, right? This is the early Earth. The early Earth was much different then than it is now. My goodness, the early Earth is different now than it was, or the Earth now is different than it was 10,000 years ago. So anyway, the early Earth, the atmosphere, number one, was a really nasty one. There were a lot of volcanoes going off. And folks, it's not like volcanoes today. I mean, there were volcanoes around today, but in some cases, like we'll look at later on, there were these huge areas of Earth, like out in Siberia, by the way, huge areas of Earth that were just like these lakes of lava and all this steam coming off and all these really nasty gases, hydrogen sulfide, and these things that were really bad for life. When we talk about it later on, and specifically we go to Siberia, the example I just used, that was bad for life. That actually killed off a bunch of life. But here we are in the early Earth. So you can see how life is being limited by these nasty gases and just filtering out the light. Just a horrible place. You wouldn't build your house where there was a bunch of volcanoes going off. There were these hot pools of water. Everything was very warm, and very often things come running into the planet. Comets and asteroids, and bam, it's a pinball gallery. And gosh, even if life begins, because they even now think maybe life began more than once. Even if it begins, it gets hammered pretty quick, okay? The important thing to understand about the early life is this is the way it would have been, right? Along with the volcano eruptions that were going off, there would have been this interesting chemical thing about the early atmosphere. See, the early atmosphere was reducing. So now you got to go back to Bio 1. Remember the words oxidation and reduction. Oxidation, right? Oxidation is when electrons are removed, or in a very larger sense, it means when something's being torn down. When you tear down molecules when you eat in respiration, you engage in oxidation. You're tearing down that molecule. You're releasing energy from it, but you're tearing it down. And the opposite of that was reduction. Reduction was specifically when you put electrons onto an atom, or an element, or in the bigger picture when you're building something up. Photosynthesis, that part where it takes carbon dioxide and makes food out of it, you make a simple molecule into a complex molecule, which requires the energy from the sun. That is chemically reduction. All right, now the early Earth's atmosphere had really no oxygen at all. So it had a reducing atmosphere. It had an atmosphere that was actually conducive for making bigger molecules. Okay? The reason why remember I talked about in Bio 1, the reason why the paint on your car deteriorates with time, gets that nasty, is because it's being oxidized by the oxygen in the air. It's about 20% now. But back then, when there wasn't any of it, the atmosphere was actually different. It was conducive for making things bigger, polymers. It was conducive for going simple to complex. Keep that in mind. So now it's at the chemical level. Okay, but we're gonna make that jump, that more or less that jump between chemistry and biology as we move through. But let's go at the chemical level. All right, now in order to make something bigger, in order to engage in reduction, you need energy. That's the law of thermodynamics. Those laws of thermodynamics and physics and chemistry were all around long before these laws of life came around, and so they had to obey all of them. I know you've heard me talk about that too. So where did the energy come from? Well, number one, it came from the ultraviolet light. Ultraviolet light strikes the Earth today. It comes from the sun. It came from the sun back then, but there was a difference back then as now. Ultraviolet light, much of it, oh, thank goodness, a whole lot of it is absorbed by the oxygen in the atmosphere, specifically a type of oxygen known as ozone, which is 3 atoms of oxygen. O2, that 3 is subset, obviously. Ozone is a special type of oxygen that blocks out. Why do I say luckily? Because UV light is really bad for us, folks. It tears apart our DNA molecules. It creates skin cancers. It creates lots of problems for living things on Earth. In high ultraviolet light, we wouldn't be able to survive. Some of it does reach the surface of the earth. Most of it is filtered out by this special type of oxygen. Not around back then. Not good for life, but a source of energy. Lightning, too. When volcanoes erupt, okay, what creates lightning is friction, folks. In a water storm, or in a rainstorm, the updraft and the downdraft of the air scrapes molecules against each other. So just like you take the old thing where you put the balloon against your hair and you get the static electricity, that friction, that scraping is creating electricity. Air is moving up in a thunderstorm, and then it's falling down because it's moving up because it's warm, and then it falls down because it's cold. But on the edges, they scrape and they make lightning. Right? Now, that can also happen, by the way, in a volcanic eruption. The cloud coming out of the top of the volcano is hot volcanic air coming up with a lot of rocks and little particles and all kinds of things in it, and they're scraping against each other, and what they're doing is they're causing friction, and that friction is causing lightning. Look online. Let me go find their website because it's so beautiful if you've never seen it before. It's lightning, volcanic lightning. How to spell? There it is. I'll send you a link to a picture. All right, here we go. Volcanic. Yeah, let me get you the link. Click on the link. Here we go. Yeah, click on that link, and that's a picture of volcanic lightning. Awesome. Same reason, though. Same reason. But anyway, lightning is a source of energy, right? What is a lightning bolt? 100 million or 100 billion volts or whatever it is. I don't recall, but that's definitely a significant form of energy. Other forms of energy may have come up things like geothermal vents. One of the other places that life may have began, or one of the other contributors, or maybe the source, or maybe just part of the source, was these underwater geothermal vents. Think about volcanoes underwater, deep underwater. These we didn't know these things existed until, what was it, the 70s? Yeah, or the 80s when they discovered them. The first ones discovered off the west coast of South America, I believe, near Galapagos, as a matter of fact. But geothermal vents, there's a real special thing that's going on down there. It's basically, like I said, a volcano underwater. And there are some really bizarre life forms down there, folks. There are some life forms down there that exist in a small little spot around one of these little mini geothermal volcanoes that exist only there, only around that little tiny volcano. And when that little tiny volcano goes extinct, because volcanoes go extinct, like the one on the right on the picture, all them critters die. And those critters are found no place, they're endemic. They're found no place on Earth but that little tiny place around that volcano. And they're life forms that we see no place else. And they make, and the way they make their living is they make their living by doing a type of metabolism that's very bizarre. It's kind of like photosynthesis, but it's a really bizarre type of. Now, I keep looking ahead to see, don't I talk about these guys later on? I know I do, or I might talk about them in some other chapter. I think when we get to the prokaryotes, I talk about them, okay? So I'll talk about the geothermal vents. But anyway, that's another place where we could have started getting this polymer formation. So again, volcanoes underwater is basically what we're talking about. Okay, the last one is really kind of cool. Hot sand, hot clay, hot rock, hot surfaces. In an early Earth, there would have been a lot of those. A lot of lava, a lot of hot surfaces, right? So, all right, so there's plenty of places for the energy to build these more complex organic molecules. But now we're going to move on to the point where we kind of make that jump between life and non-life, all right? Let's go back to the beginning where we look at this organic molecule formation. All right, so we get the energy from this kind of stuff on Earth, but what about the other ingredients? Well, the first thing that I want to actually introduce you to is I want to show you how we refute one of the more common theories that was around for where life came from, called spontaneous generation. Spontaneous generation is that life pops out of non-life. So in a weird kind of way, I'm going to first disprove spontaneous generation, and then we're going to talk about how life came out of non-life. You remember, it's the mechanism thing again, right? Darwin's theory of evolution, Lamarck's theory of evolution, the mechanism was wrong. The idea that organisms change through time was not wrong. He wasn't blind. He could see it in the fossil record. So when Pasteur refuted spontaneous generation, he didn't refute life from non-life. He refuted the mechanism. The mechanism was spontaneous generation. All right, in the next slide, I'm going to show you another more plausible mechanism. But let's talk first about good science, man. Good science. Louis Pasteur, the guy that pasteurized milk, right? Helped pasteurizing milk. All right, here's what Pasteur says. Back in that time, it was thought that life came out of non-life, that if you put a piece of meat on the side of the road, and that piece of meat is non-life, it's a dead animal, that when you came back and there were maggots on it, that the maggots came out of the meat, that somehow the non-living components in the meat, the seeds were there, and boop, life popped out, and we had the non-living meat giving rise to the living maggots. Now, we all know that the maggots came from the flies that laid the eggs that the guy didn't see until they became maggots. All right, we all know that, but that's the theory, right? That's like Aristotle. That's the thought. So here at the first top, here's what Aristotle. Here's what Pasteur does. He takes a broth. A broth is it's something that bacteria would grow on. Okay. All right. If you left soup out long enough, it would get spoiled. So anyway, he takes his broth and he sterilizes it by heating it, which is what doctors do, right? When they put things in an autoclave, they use heat and pressure to kill living things. But he makes the broth sterile by heating it, boils it, right? If you're gonna boil your water, leaves it out, and guess what? It spoils. Okay, now spontaneous generator folks who would subscribe to that theory, well, they would say that the organisms in the broth, the microorganisms, because, back then, remember with microscopes, you didn't really know where these things were coming from, these microorganisms. So they thought, well, they came out of the broth. That even though you sterilized the broth, there were some ingredients in the broth that once you let them sit, came together, and boom, this non-life, the life thing happened, and that's where the microorganisms came from. Again, they don't have really good microscopes back then. So some of this has to do with technology. All right, so here's Pasteur's setup down below. Here's what he does. First, he takes the broth that he sterilized, and he seals it off, and no organisms appear. Good control. Because now there's nothing in there at all. There's nothing. What Pasteur has done is he's made the environment only the flask. So, you spontaneous generators, if life comes from non-life in this environment of nothingness, this flask right down here, in this environment of nothingness, then your organisms should appear. Okay, they don't appear, obviously. Nothing grows there. It's sterilized. It's sealed off. We know where the microorganisms come from, but guess what the spontaneous generators say? They say, well, the nothing is the problem, Louis. Maybe they said it with a French accent because he was French. It's the nothing. What do you mean, the nothing? Well, when you sealed it off, you prevented the ingredients that are going to make life from being exposed to the outside environment. And there's something in the outside environment they need. It could be the oxygen or something out there, right? Okay. Of course, we understood that oxygen was important for living things. So let's say Pasteur was going to entertain their thought of the oxygen. He makes the flask over on the right now, the one in panel 3. He makes it with a gooseneck, a swan neck. It's like the trap on your sink. So things a little heavier than air. They don't have to be too much heavier, just a little heavier. Just like your ring, if you dropped it down your sink, would get caught in the trap. Okay? I don't know why that's supposed to be an arrow, but they would get caught. So microorganisms, by the way, weigh something. They weigh more than air. Probably, proportionally speaking, they weigh a lot more than oxygen. So guess what is going to happen? You can expose the flask to the air, but keep out little tiny things. Doesn't mean back then, because the microscopes weren't very good and we couldn't hardly see these little tiny things, there were people like Pasteur that were postulating, they were hypothesizing that the reason why it appeared that spontaneous generation worked, because we couldn't see something. We thought that the maggots appeared out of the meat because we didn't pay attention to the fact that there were eggs. If we'd known there were eggs, then we said, oh my goodness, something laid the eggs. When Pasteur connects up the swan neck, he says, all right, guys, all the air is there. The only thing the swan neck is trapping is the things that I think are there, these microorganisms. And guess what? Nothing grows. Now, the final step he does really cements Pasteur's work, because he breaks the top off and he says, okay, now everything from the outside can get into the inside. And guess what? The broth becomes spoiled. All right. So why did this refute spontaneous generation? Pasteur, once again, demonstrating the idea that it is something that's coming from the outside that's causing the broth to spoil. The broth doesn't grow in a nothing environment like in panel 2. It's got to have something from the outside. It's not the air, by the way. It's not the non-living components out there. It's something out there that's giving rise to it. There's a living thing out there, folks. A small one. You can't see it, but you can see the evidence of it. Now, you know, microscopes soon become better, and they continue to support Pasteur's theory, or they continue to support Pasteur refuting it. Okay, I just got a message that says my connection quality is bad. So if you're having problems, then I'm sorry, but I'll continue here. So you see how he refutes it? He refutes it by showing, listen, everything that you say is there for these spontaneous generation things to happen, and it doesn't until I open it up and tell you that I think there are small little things out there. Okay. So it wasn't the mechanism of spontaneous generation that gave life from non-life. But let's kind of take a look at the mechanism. We're going to look at the mechanism in some steps, all right? So out in the lobby of the Science and Math building, there's a mock-up of this sitting in that display cabinet on your right. And what this is in 1953, a couple of folks, Miller and Urey, I don't know their first names, they came up with something that we call the Miller-Urey experiment. And what they did, oh, there's my picture of the geothermal vents. Okay. What they did is they tried to simulate in the laboratory what the early Earth was like. So if you look in the upper right where the big ball is, there's the atmosphere, which they filled with things they thought were in the early Earth's atmosphere. As a side, if you ever wonder how they do that, is bubbles of gas get trapped in ice. And the glaciers are kind of like the fossils. The further you go down in a glacier, the deeper you go, the older the ice is. So the little bubbles of air that get trapped in ice, and you can even see in your ice cubes when you put them in a drink sometimes, that air is a sample of what the air was like back then. So if they can kind of correlate places, and the air also gets trapped in rocks, so as the rocks are cooling off from lava, they will trap these little bubbles, and they're porous. So with these little micro analyzers that can tell what the early Earth's atmosphere was pretty much like. And it was reducing. It had water and nitrogen and ammonia, carbon dioxide and monoxide, a bunch of methane, all hydrogen, all things. Hydrogen's very abundant. 99% of everything out there is hydrogen. The water and the carbon dioxide and the monoxide and the methane, all that is very common. Even today we get them out of volcanoes, all right? The electrodes in here are going to stimulate lightning, so the energy that's involved. And if we run this whole experimental setup many, many times, around and around and around and around, so basically what we're doing is we're boiling water in the left, which is going to be the ocean. So we're providing the water in the atmosphere. We're providing the energy. The water cools off. It rains, right? And down here at the bottom, look at what they found. Down there at the bottom, when they let something like this, now look at those molecules in the reducing atmosphere. None of them are complex organic molecules. The other one even strikes as being an organic molecule. The other one just barely gets in is methane. Methane is an organic molecule by definition, right? But it is not a molecule that is used in terms of greater or bigger life forms, right? Methane is usually something that many organisms give off. But down below, there was collections of small organic molecules, more complex than methane. Still, we're not talking about starches, but more complex than methane. But there were even some amino acids as well. All the components are there to make amino acids, nitrogens and carbons and all that sort of stuff. So some very, very, very simple molecules that we normally associate with life. Right? Remember back in Bio 1, we said that amino acids were the building blocks of life. So, and this has actually been created in some simpler, even high school laboratories. And if you let it run enough, you do. You get a little collection down below of some relatively simple organic, maybe even some amino acids, okay? All right, now this might have not happened in the atmosphere. This goes back to the geothermal vents. There's a picture of a geothermal vent. There are hot ones and cold ones. The hot ones can be, oh gosh, 500 degrees. The water doesn't really even boil down there because of the pressure from the water. So that column you see behind there is this column of, just hydrogen sulfide, carbon dioxide, all those things that were in the volcanoes. So even though this, we may have kind of a variant of the Miller Urey, maybe this didn't happen in the atmosphere, maybe it happened down there. But now, as I said, there's a lot of theory out there that say maybe it happened in more than one place. Maybe down in the geothermal vents and in the atmosphere, maybe throwing a little panspermia. It's fascinating, right? Maybe life began more than once. Who knows? Okay, so now we're making simple molecules. How do we link them together? Well, in order to link simple to big, in order to go into a dehydration synthesis, in order to make a more complex molecule, a simple one, you need energy. There's plenty of energy around. Heat is energy. It's not a very efficient form of energy, but it is a form of energy. You can use it to cook your meals. So if we had this soup, imagine the ocean with all these relatively simple organic molecules and maybe some amino acids, if they splashed up onto these hot surfaces, maybe not on the lava might have been a little warm, but the rocks around or the sand or the clay, the heat of those surfaces might have been enough to stick those molecules together. Okay? So we're moving into the macro molecule. We're moving beyond methane now into the more complex organic molecules. We're still not really at life. You might be disappointed at the end of this. We still kind of stop short of considered something alive, but we get pretty close. But at this particular point, we're linking together organic molecules, okay? So we've made organic molecules, simple ones. We're now linking them together. All of this happening in the soup of the ocean is nice, but as we all know from our discussion about chemistry, especially in Bio 1 or enzymes and substrates, things get together by randomness. Enzyme and a substrate don't recognize each other and seek each other out. They bump into one another. And I think I used the analogy in Bio 1. If you were in the dark, you and another person, you'd have a much better chance if you're walking around trying to find each other if you were in a phone booth than you were in a warehouse, right? If it was a smaller area, your random wandering, because remember, it's dark, would be more efficient if you were in a smaller area. Makes sense, right? So we got these reactions occurring. We got these reactions where macromolecules are being made and all this kind of stuff, and we might actually increase the rate of those reactions if we package them in a smaller area. Well, there just so happens to be a molecule that is abiotic. So it's a molecule, it's not created by living things. Okay, there's a abiotically produced molecule, very similar. I think it's actually quite similar to these molecules back from Bio 1. You remember those phospholipids? Phospholipids are abiotically produced molecules. All right, they're organic, but what would have happened back in time if a bunch of phospholipids got together, especially in a water world, in a world full of water? Remember how they react in water? They make a spear. They make maybe something we might call a liposome. You can make liposomes in the laboratory. It's not a living thing at all. It has a selectively permeable bilayer membrane, just like phospholipids. It doesn't have all the proteins embedded in it, but remember, some things go through the phospholipid bilayer. Liposomes can bud off, so reproduction's in quotes here, so we can get, a package, you see, that is separate from the outside world, that is, right, that phospholipid bilayer, that has the ability to make copies of itself, so kind of a reproduction. So let's see, on the right-hand side, what we're going to do is we're going to suppose a few things. So we're going to take a little scientific license here, right? We're going to introduce to the environment with this protocell. The green thing is the protocell. We're going to introduce some glucose phosphate. Those are organic molecules, so perhaps during this organic molecule formation, something like glucose phosphate or something similar to glucose phosphate was around, and the cell is going to take that up. Now, if we add a couple of enzymes, if we do that in the laboratory, those are both enzymes, they end in ASC. But if we assume that maybe some relatively, I don't know, simple types of molecules had the ability to act like enzymes. Like a little bit later on, we're going to talk about that self-replicating molecule at the very end of all this. And some of those self-replicating molecules might have actually, maybe you don't call them enzymes, but they're enzyme-ish. So if there was a phosphorylase type enzyme and an amylase type enzyme, what all of this is showing you in that green circle there, if you take a protocell and you assume some relatively simple assumptions that there might have been some organic molecules forming, you can actually get metabolism. That's what this is all about, folks. This is metabolism. Glucose phosphate gets turned into starch by sticking them together. Starch gets turned into maltose by tearing them apart. And when you do that, whenever you go from starch to maltose, you release energy. So we have this energy thing going on. We have this ability to store up energy-rich molecules, and this ability to release that. Now, that's making some assumptions, no doubt, not out-of-the-world assumptions, but it's making some assumptions about what the early Earth was like. And it's saying that what we're seeing here is at a very simple way, at a very low level, is some metabolism. Remember, metabolism is your chemical workings of your body. So when you take in food and you release energy, or even when plants photosynthesize, there's nothing magical about that, right? It's chemistry that's doing all that. So we're just saying, could chemical conditions been in such a way, that we could get this simple metabolism? Some interesting evidence about the formation of liposomes comes from what we knew the early Earth was like too, with all the volcanoes going off. We knew there was a lot of this, it's a really tongue twister of a word, montmorillonite clay. I tried to underline it. Montmorillonite clay is very common from the weathering of volcanic ash. Remember, in the early Earth, there was a lot of that. And what we find in the laboratory is as we add more and more of this clay to a mixture, let's say a beaker or whatever, we get increased formation of these precursor molecules. What we mean precursor, the molecules that will eventually become the liposomes. So maybe those phospholipids that we're talking about. So if we assume there was a lot of this montmorillonite clay back in the early Earth, and there was lots of volcanoes, we have lots of evidence for that, then, we might have had an environment in which these abiotically produced molecules that made this liposome could have been there, right? All right, now, with all these molecules and these things going on, we now have a situation where if we could come up, or if Earth came up with some relatively special type of molecule and then packaged it in these protocells, we get pretty close to something alive. Maybe not quite alive yet, but pretty close. So let's look at the fourth thing that we think might have happened. And again, this could have been something that happened on Earth, or maybe these molecules were brought from someplace else, or maybe the seeds were, or whatever the case may be. Number 4 in biogenesis says that some type of molecule that could make copies of itself had to occur. You remember, one of the characteristics of life was reproduction. Life can't perpetuate, can't live forever. It's a system. It falls apart. Life can't perpetuate unless it can reproduce. You can reproduce asexually or sexually or, whatever way. But without the ability to reproduce, maybe some of those early life forms, maybe that's what they did. I wonder sometimes if life began, but somehow the characteristic of reproduction didn't occur, and then it died, and I don't know, maybe it was a number of starts before this self-replicate. Now, why do I keep focusing on self-replicating? That's what DNA does when mitosis happens, right? It even does it in meiosis as well. You make copies of DNA. When you do transcription, messenger RNA or DNA to messenger RNA, that again is copying. Right? You're copying the code. All right, so let's imagine what has been hypothesized back 3.5 billion years ago as a world dominated by RNA, an RNA world hypothesis, right? For one thing we've talked about, you got to have metabolism for life, you got to have inheritance. Inheritance is that self-replicating molecule. You need that for evolution, don't you? Without this self-replicating molecule that can be changed and can be somehow affect the thing, the phenotype, you're not going to get evolution. Well, back many years ago, because some of them, many years ago, they even exist today, there are these things called ribozymes. This in the upper right is a really cool one. You can see these are all the bases of RNA. This is the hammerhead, I guess because it's got two parts to it up at the top maybe. Hammerhead ribozyme. What's a ribozyme? Well, it's two words, right? Ribosomes, and enzymes. So it's not an enzyme, okay? So it's not an enzyme, but it's a sequence of RNA that couldn't do something like enzymes. It can catalyze reactions. So maybe it wasn't amylase and phosphorylase like we know it today, maybe, that did this, right? Maybe it was, maybe it was some ribozyme type of thing that was able to carry out these reactions. And RNA was actually capable of self-reproduction, you see. And the reason why it was capable of self-reproduction is RNA was creating these ribozymes, and these ribozymes were allowing it to have this ability to process energy. So we're just kind of there, aren't we? Not exactly maybe living. You wouldn't say a proto-cell with some ribozymes inside and some RNA doing, that reaction from a couple slides ago. You might you might say, I don't know if that's life yet, but we're definitely starting to fill in all of the criteria, organization and metabolism and all that sort of stuff, right? Okay, any questions on any of that? I spent quite a while on that, but now once we have a decent idea of how life began or must have began and the progression of life, what happened from 3.5 billion years ago to today, what's going on with the rest of the movie? Well, for one thing, we know the dates when these things happen because of we dating the fossil record. We've talked about dating the fossil record before, and in dating the fossil record, what you really do is, in some cases, you can date the carbon-14, but that's only back to about 75,000 years. So going back later than that, we have to date the actual rocks the fossils are found in. And remember, what's going on in this situation is we say that organisms bring in carbon all the time, right? That's part of what we do. That's what we need for. We need that element. Most of that carbon is carbon-12, but a little tiny bit of that carbon is the isotope carbon, carbon-14. So when you die, you no longer bring any carbon anymore. So that carbon-14 begins to, radioactive elements begins to disintegrate. We know at the rate at which it does that half-life. Same thing with potassium-40 and uranium-238. They work the same way. When the rocks are cooling, once the rocks have cooled, the amount of radioactive potassium-40, uranium-238 is locked in that rock. Once that rock goes from lava to rock, that amount is locked. And from that point forward, it decays, and we know how fast it takes to do that. So we can take all that, and we can build this really cool picture, which I'm going to go through relatively quick here, because I'm going to go through it in a little, the rest of the class, actually. We're going to spend a lot of time talking about the rest of what happened. So we're down here at the bottom. Origin of the Earth, 4.6. Oldest known rocks, 3.8. Life, somewhere in there. 3.5, 3.8, 3.9, not really sure, but something about 2.7 billion years ago happened that changed the Earth considerably, because before that, the Earth was reducing, remember? But after this, the Earth became oxidizing. Now, oxidation is not good for molecules necessarily. It tears them apart. But for living things, oh, man, we love oxygen. Oxygen is really good because it allows us, instead of getting 2 ATPs, take it all the way back to bio 1 again, instead of only getting 2 ATPs from just glycolysis, if you got oxygen, you're getting what, 32 to 36? You get significantly more ATPs if you respire with oxygen, with oxygen than if you respire alone, don't you? Without oxygen. All right, so that changes everything. So we'll run through all of these, but we start to get eukaryotic fossils. We start the algae. There's the Cambrian explosion. After that, holy cow, Katie barred the door. A lot of weird critters in the Cambrian that we might not recognize. We've seen some of those. I have some fossils of those. And then soon after that, we start seeing some stuff that we might still recognize. Look at that. There's the horseshoe crab. Horseshoe crabs are what we call living fossils. They're still alive today, but they haven't changed much since 450 million years ago. Vascular plants, which we'll talk about, bony fish, tetrapods and insects. The earth gets full of forests during the Carboniferous. That's why we call it that. That's where most of our coal deposits come from. So the earth was lush back then, man. I tell you, it was a tropical paradise. There was all kinds of trees and all kinds of things to eat, and seed plants started evolving, because initially it was just ferns and mosses, and they don't have seeds. But most of life was living in these wet areas, and then the reptiles came along, and they figured out a way to live in drier areas. And there were plants that were learning how to do that as well. So living on the coast or living in these wet areas, that was relatively easy. Like, for example, during the Carboniferous, the amphibians, frogs and toads. But we all know this thing about amphibians. They have to go back to the water to lay their eggs. So they got to be near water. I guess you can't hop too far if you're a frog or a toad. But reptiles lay a different kind of egg. So guess what? Reptiles said, well, I don't have to stay here just in these water areas. I can live in water areas, and I can live in drier areas. So just about that time, look what starts happening. Just about 250 million years ago, we say the reptiles start radiating. They start becoming many species, speciation, punctuated equilibrium, because there was a big extinction event that occurred back during this time as well. But the reptiles really start going crazy in the number of species. Insects do too. It's all about these different environments. And for reasons that we'll talk about later on, it wasn't good for everybody, though. Some organisms began to die off. Anyway, after 250 million years ago, this is the dinosaur era. Right? That's when the dinosaurs lived, about 200 million years. They died off during an asteroid strike 65 million years ago. But notice throughout very quickly that other things were still evolving, even mammals. Mammals originated back at the very beginning of the dinosaurs. It wasn't like dinosaurs died off and then the mammals. What happened is right after the dinosaurs died off, then the mammals radiated, like the reptiles did. So that asteroid strike was very fortuitous, wasn't it? All right, flowers and plants of different types. And then, of course, we move into this group, and a lot of stuff was going on after the dinosaurs, especially among the mammals and the flowering plants. But once that pressure of the dinosaurs was released, then Earth kind of went through this radiation of many different mammals and angiosperms, and we come along, and then some of our ancestors come along, and here we are at the end of the movie. So we're going to go through each of those in a little more detail, but it's kind of neat to look at that from the very beginning, right? So we go from some of these early prokaryotes in that 300 billion years, 350 billion years or so, to all the diversity. And remember, what we're seeing is only the things that survived. Most things didn't survive. Most things went extinct. Okay, so that's a nice overview. Let's go back to the beginning again. Let's kind of see what the early Earth was like back. Okay, we got life, we got biogenesis. We don't have any real clear fossils before about 3.8 billion years ago. There might be something back then, but the early Earth was changing. There was a lot of tectonic activity. Fossils end up being encased in rock. So if that rock gets recycled back into the Earth and gets melted again back into lava, those fossils are gone. That's why sometimes you might read online or in the news that they found this old rock. Like maybe they found rocks in Greenland that were 3.2 billion years old. What we mean by that is those rocks have never been recycled. They formed 3.2 billion years ago. They've never been turned into lava again. So whatever's in there, whatever fossils in there was in there 3.2 billion years ago. Okay, so no clear fossils before about 3.8 billion years ago. So on the clock here, we're right there. We're getting ready to move into this area where we start to see some evidence of organisms that survive. But just prior to this point, nobody knows exactly how long it took for this to happen. But the early Earth, like I said, was not a good place. Asteroids and meteors and all that sort of stuff. There was water, but not a lot, because all of that asteroids and comets and all that stuff bouncing off the Earth, evaporated. A lot of heat going on there. Uh, but about this time, 3.8, 3.9, the pinball gallery begins to settle down. We still have remnants of this in our solar system today, too, by the way. The asteroid belt between us and Mars is remnants of those rocks that never quite coalesced and made a planet. And then out way beyond the solar system, there's another set of rocks and comets. And again, those are things that never, they're just pieces left over, if you will. Anyway, when the bombardment ends, the Earth begins to cool, liquid water begins to form, and we know, of course, that in order for life to occur, Earth life, carbon-based life, you're going to have liquid water. So that's when the prokaryotes begin their thing. All right, the bacteria, the archaea, those kind of critters, about 3/4 of the Earth's history, by the way, is dominated by the prokaryotes. And very early on, evolution went to work. Very early on, evolution went to work at the grandest scale because we separated those two prokaryotes very quickly into two domains, the archaea and the bacteria. I like to call these guys the extremophiles. There are some bacteria that can live in extreme conditions, but the archaeans, they're known for that. They're known for living where it's really hot, really salty, really places where you just normally wouldn't find life, which if you think about kind of fits in what we thought the early Earth was like, right? But again, there were a lot of bacteria back there in that time too. And then they have dominated the Earth ever since. The dinosaurs came and went, but bacteria have always been there. There's more of them than there are us. Now, as far as metabolism went, some of the early prokaryotes were chemoautotrophs, okay? Some are chemoautotrophs. Now, that's similar to down right, or in lower right, where I wrote photoautotroph. Photoautotroph, folks, is the fancier way of saying photosynthesis. Same thing. Right? So before photosynthesis, or maybe we can say at the same time as photosynthesis, some of these early prokaryotes were not photosynthesizers, but chemosynthesizers. They did chemoautotroph. They used chemical means by which to make their own food. That's what autotrophs do. Plants are autotrophs because they autotroph. Troph is to feed. They make their own food. Plants do it using light energy because you need energy to make food because you're going simple molecules to more complex. But in chemoautotrophs, they use chemistry. Like, for example, one of the things they do is they can actually take carbon dioxide and hydrogen, and the act of that sticking together to make that methane, they can actually get energy from that. Some can oxidize sulfur. So whenever you oxidize something, you release energy. Some can oxidize hydrogen sulfide, that stinky rotten egg gas. Very common in volcanoes, by the way. Sulfur also. Iron. Some early prokaryotes could actually live on iron. That we didn't really know much about this until we found the Titanic. Till Ballard found the Titanic a few years ago. And that critter in the upper right, that little one, is a bacteria organism that lives on the Titanic. And I believe it's only found there. We'll talk about it more later on, its name and such. But it actually is living on the iron plates, or the iron in the plates of the Titanic. So what I'm trying to say here, if the early Earth was a rough place in terms of getting food, these early organisms have some pretty unique, relatively chemically simple ways of getting some energy, right? So these organisms were able to take the oxidation of sulfur, or the oxidation of hydrogen sulfide, or the oxidation of iron, that energy, and use it in place of sunlight. All right, so all those processes of oxidation are releasing energy. Now, they're not using that energy necessarily for themselves, but they're using it to make more complex organic molecules, just like the photosynthesizers do, right? Photosynthesizers take carbon dioxide and water, and they link them together to make glucose, to make food. That's what we're doing here. We're oxidizing the weird, bizarre things and using that energy just like the sun is being used by plants. Now, speaking of photosynthesis, let's take a look at that for just a second. This is an interesting sort of evolutionary question. Photosynthesis, if you remember from Bio 1, that was some pretty intense stuff, right? There was some pretty, lots of different things were going on, and it was real easy to get lost in the details, right? So photosynthesis is a complex topic, no doubt. If we were looking at the evolution of photosynthesis, and we had this very simple sort of tree we were looking at, there are two hypotheses to how organisms are photosynthetic today. And the one on the left says that the ancestral prokaryote was not a photosynthetic critter, and photosynthesis evolved later. So heterotroph first, photosynthesis later. And the other hypothesis on the right said it was the other way around, that photosynthesis evolved first and that it was lost in some organisms. So heterotrophs were ancestral organisms that could photosynthesize. In science, overall, in all of science, we're always looking for an answer that is the most parsimonious. Now, the word I just said is parsimony. Parsimony says that the simplest answer to something, the least complicated, is usually, not always, usually the right one. And that idea of simple is relative, of course. But if we looked up at night on the beach and we saw bright light in the sky, we could postulate or hypothesize a few things. Aircraft coming in for a landing, I want to say that one last, a helicopter, a star, a planet, a UFO. One of those hypotheses is less parsimonious than the others. One of those hypotheses, remember science, you never prove or disprove anything, but one of those is least likely. I'm not saying I don't believe necessarily in UFOs, but if I saw light at night, that would be my last guess. So between these two, keeping in mind that complexity of photosynthesis, we believe what happened if we apply this Occam's razor. Occam's razor says keep things simple. We believe that photosynthesis was the norm. Photosynthesis began first, and that organisms that became heterotrophs, like the line that led to us, we lost the ability to photosynthesize. We became heterotrophs. Now there would have been advantages to each each of those, or they wouldn't have happened that way. Then you could probably think about some of them. You're a photosynthesizer. You are limited to sun. So if a volcano goes off and you don't get a lot of sun for a long time, you're going to have problems. But heterotrophs, even those who eat plants or eat things that eat plants, can at least move someplace where there is the sun. Okay, but let's go back to these photosynthesizers for just a second. It was ancestral the ancestral prokaryotes were photosynthesizers. Let me jump back a couple slides because I want to show you where we were in all this. And that big picture I was trying to show you earlier, remember, that's right there. So we're just about this time where we get these early prokaryotes beginning to diverse and beginning to increase in abundance. And in those little bubbles of rock and ice, look what we found. We found that oxygen levels began to rise, like really fast, too, by the way. Is my pen going to work? Yes. They begin to rise like that. They begin to go up exponentially. So the oxygen in the Earth's atmosphere, which is, what, 20% today, back then accumulated very quickly. And that's because there were a lot of these photosynthesizers around. There were a lot of these cyanobacteria like Anabaena and Oscillatoria around 2.7 billion years ago. And these things were photosynthesizers. We're going to look at them underneath the microscope when we get in lab. But these things do the same thing that plants do, and they produce oxygen. And there were lots and lots and lots of these Anabaena and Oscillatoria. They made these huge mats, huge mats. Down on the right are stromatolites, and that's on the edge of the ocean. So the ocean has eroded between those to make these columns. But those columns are columns of cyanobacteria. And what they do during their process is they produce these mineral compounds that form these stromatolites. But they build them up from the bottom to the top. So you can see is they built these huge columns, and the ones down below are dead. That's just the mineral part. And the ones at the top are still alive. And the ocean is eroded in between them. But what I'm trying to express here is just how much, man. There were huge mats of these things all over the Earth, making lots of oxygen. The oxygen initially doesn't do anything to the air because in the water there's a lot of iron, so this chemical thing goes on. It's called rust. So first we're going to get iron oxide, but then after a while we saturate, and just like when you open up a Coke, the bubbles come to the top. If there's too much gas in the atmosphere, after a while it bubbles up. If there's too much gas in the water or liquid, it bubbles up into the atmosphere, right? So we get this dramatic rise, relatively speaking, in terms of time of oxygen. And oxygen was good for those of us our lineage that became aerobic respirers, right? We could get 32, 36, 30, 38 ATPs out of a glucose, where if you don't have glucose, you just get 2. So having more energy means we could get bigger and stronger and more complex, right? Good for us, not so good for some organisms, though. There are some critters out there, even today, even today they're alive, that are called obligate anaerobes. In other words, they die in around oxygen. Oxygen, remember, removes electrons off of things. Now, we figured out a way to deal with that, at least for 75 or 80 years. But back then, these organisms, never having been around oxygen, once oxygen levels rose to a certain level, they just begin to die off. Now, in some cases, some of these prokaryotes that were dying off, they entered into these symbiotic relationships. Symbiotic relationship literally means two or more things living together. Clownfish and anemones is a symbiotic relationship. We'll talk more about those later on too. But some of these prokaryotes actually entered into what we call an endosymbiotic relationship. They actually became they actually were able to survive by ending up inside of another cell. So I'll come back to that in a moment. But again, so we get this rapid rise 2.2 billion years ago. So 2.7 to 2.2, half a billion years. Heck, that's a long time, but we get this rise of oxygen, this relatively rapid rise. So now we can start making now the Earth can start producing not just prokaryotic organisms, but eukaryotic, something a little more complex, because we have this rise in oxygen. We have this additional source of energy. We get a lot more ATP, right, if we're using oxygen than when we're not. You can see it on this graph here. Look at time billion years ago, 2.7 it begins. And at about 2.2, we see this rapid rise. Now, this is stuff that's going on in our modern day history, of course. But what that did for biology back about 1.8, 2.5, somewhere in that vicinity, this doesn't happen overnight, is the O2 levels begin to rise. And like I said, that was really good for those of us that could take advantage of that. But for other ones, they began to take off. They were on the run. They probably moved down into the dirt a little bit, but oxygen does penetrate into there. They might have moved into the water, but there was already oxygen in there. But the point is, a lot of them begin to die off, but not all of them. And this is really kind of cool. This is something called endosymbiosis I just mentioned. Some of these prokaryotes survive through endosymbiosis. It's a glimpse. It's a glimpse into the past and something that's alive today. Let me show you what I'm talking about. This is so neat. Wait a minute. Yeah, let me go to this and I'll go back to that slide. It goes out of order. Anyway, up in the upper left, we have some ancestral prokaryote. It doesn't have a nucleus. We know that. They have nucleoid regions. Now, this ancestral prokaryote began, or some version of them, began to be very good about engulfing, invaginating, it's called. They're very good. They begin to do it a lot. So they begin to engulf things. And one of the things they would engulf would be food. You remember endocytosis, right? So that would have been a good thing. But everything in a cell is made of membranes. I know you heard that a bunch of times. And all these membranes begin to engulf. And they form, like, for example, the nuclear envelope and the endoplasmic reticulum. And I spent a whole Bio 1 talking about the importance of membranes. So I'm not going to do that here. I'm sure you know that. But they also begin to engulf other things. Like, for example, some of them engulfed a mitochondria, right? Some of them engulfed a mitochondria. And mitochondria actually is a relative. The organelle, the mitochondria, is a relative of a free-living thing that we think existed back then, some sort of cyanobacteria. There's a fossil of one. Now, there are cyanobacteria around today. They're the photosynthesizers. But the one that were originally our mitochondria were cyanobacteria that were aerobic, all right? So they were able to use the oxygen, all right? So while in some cases endosymbiosis saved prokaryotes from dying off, in some cases certain prokaryotes were able to use the oxygen, but guess what happened? They were having difficulties dealing in an oxygen environment. So aerobic bacteria were getting the extra energy, but the oxidation of the environment was causing them problems. So they became engulfed by randomness, maybe to be eaten initially, but something happened, and instead of being eaten, that engulfed bacterium through time developed a symbiotic relationship with that cell. It said, hey, cell, before me, you're getting 2 ATPs for glucose. And guess what I can do for you? I can make 40 ATPs out of the same glucose. What do you think? Don't eat me, right? Now, we know that for a variety of reasons, mitochondria have their own DNA. The only things we know have DNA are the nucleus of cells of the nucleoid region. I mentioned this in Bio 1. Mitochondria look like bacteria, and they divide. They can divide independent of the cell. Okay, and likewise, some of those cells that engulfed mitochondria, because remember, plants have to respire, would have engulfed something similar to this sludge that we see up above, this green algae sludge that we might call sludge. Not a particularly nice name, but these photosynthetic bacterium, again, they were actually making it better for us, but they might have been making it better for themselves or other ones, but the oxygen was getting to them, but they became engulfed. And now you have the advantage of not only do you get a lot more energy from a glucose, you just need the sun to do that. Lots of advantages to being photosynthetic, right? You can photosynthesize, all it needs the sun. So at about 1.8 billion years ago, we start to see some of these multicellular eukaryotes coming about, right? So we get this engulfing of this endosymbiosis thing. So now we got these big advantages. Remember, it's all about evolution. We get these big advantages going on, and multicellular eukaryotes are going crazy. Maybe 1.8, the oldest ones we know are 1.2, but that depends a lot on how well those fossils preserve. But all of this increased energy, all of this more efficiency allowed for more complexity. So eukaryotic cells begin to develop more organelles and plasma reticulums and ribosomes and peroxisomes and all that sort of stuff. Advancements in the cytoskeleton allowed for better endocytosis. So now, since that engulfing was already kind of in these cells, adding that cytoskeleton, the intermediate filaments and the microtubules and all that stuff actually allowed these things to endocytosize a lot better, allowed them to bring in food, more food. So what we're seeing are organisms that are becoming more fit, aren't we? Just like we would predict in Darwin's theory. Organisms are becoming more fit. They're becoming better at exploiting their environment. They're doing things like adding a photosynthetic bacterium. A plastid, by the way, is let's just say it's a primitive chloroplast, okay? Well, a plastid is not a primitive one. It's just, chloroplast is a plastid, same thing. All right, so we get this whole idea of better, you can photosynthesize, or if you don't photosynthesize, us heterotrophic ones, and this could have been where that split occurred that we talked about, us heterotrophic ones, at least with the mitochondria, we're going to get a lot more energy. And there are plenty of these photosynthesizers around that we can eat, by the way. Plants can grow a lot, and photosynthesizers can make a lot of food for us. So boom, 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 here it happens. Everything's set. And just about this time, and I've already mentioned it a couple of times, and here it is in all of its glory, what we call the Cambrian explosion. Not so much an explosion. It lasted, well, what, 25 to 100 million years. That's a relatively long fuse. But remember, Darwin, when he looked at this, he said, geez, even at 100 years, that's pretty fast. So not sure exactly when it began, not sure exactly when it ended. I put that question mark, but I probably should put that on all of them. We have, if you look in different places, you'll have slightly different dates many times on when these different parts of the geologic history began and started. But everybody agrees somewhere around 500 million or so years ago, we get this Cambrian explosion. And what we mean by explosion is, where are we? Right about right there. So we're crawling up. I'm trying not to go through the lines. But I want you to look at the organisms that we had before and after the explosion. So before the explosion, we had sponges, we had jellyfish, and we had some relatively simple mollusks, snails. They weren't snails. They would have been sea critters, squid, those kinds of things. After the Cambrian explosion, look what we get. We get the echinoderms, the sea stars. We get the lineage that gives rise to us, the chordates, this weird group of organisms called the brachiopods, segmented worms, a huge group of arthropods, crustaceans, insects, all that kind of stuff. So basically, in about 25 to 100 million years, we went from some relatively simple, but holding on. These guys have been holding on for quite a while before the Cambrian explosion. You see that? They've been sponges and cnidarians and all that stuff been holding on for quite a while. They've been doing their thing for quite a while, and then the explosion comes along. Now, one of the reasons that we think that the explosion was driven, one of the reasons the mollusks were just about there before the explosion, what do you think that maybe drove this this explosion was the arms race? In other words, what was happening is organisms were learning to become better predators. Okay, so they were moving outside of just herbivores, and they were saying, well, wait a minute, I'm going to eat the things that eat the plants. And maybe even there were omnivores who ate a little bit of both. But the point is, when I'm being preyed upon, I'm going to try to defend myself. So what this does is this puts a very, very, very heavy selective pressure. Now, Again, I'd like to caution you. Remember, it's not selective pressure. We're not saying we're selecting against. So maybe better reworded, you could say the evolution of predation imposed a very heavy selective advantage to those who were becoming good predators and those that were becoming better at defending themselves against predators. So the prey and the predators were getting better. One was getting better at killing the other one, and one was getting better at not being killed. And guess what was happening? It was going back and forth. Predators would get good, and prey would start falling in numbers. Predators start dropping off. Prey start getting better. It was back and forth, back and forth. If you say, why didn't the prey keep getting better if the predators were dropping? Maybe the prey were eating more of the food, more of the plants, and of course their population began to drop. Homeostasis. It's all homeostasis. All right. Where am I? Oh, good. We can get into the Cambrian explosion. So Cambrian explosion, boom, we get this rapid diversification of living things. Now, what was going on land in terms of plants? Prior to this point, at 500 million years ago, prior to this point, there were plants, but they were on the edges of the land, and they were barely making on, and they were algae, and they provided food for some critters, but they were relatively simple in diversity. Moving out onto land involves something. What was going on about 500 million years ago, and I got some pictures that I'll show you in another lecture, but land masses were forming. And when land masses form, what that means, they get bigger. And when land masses get bigger, in the inside, they get drier. They're going to be wetter around the edges, and they're going to be drier in the middle. And when they got drier in the middle, that was bad for some critters, but what it did is it prevented opportunities for successful and fit ones to survive. So what was going on is plants began to move out onto land and begin to exploit these little more difficult environments in conjunction with something else, something called mycorrhizae. Mycorrhizae is a fungus that we'll talk about later. But plants, when they began to move or colonize land, and that was important because plants had to be there before the animals. And as a matter of fact, just about soon after plants began to move on the land, the arthropods started going crazy in terms of diversity about 450 million years ago. It's all connected, right? So these plants moving on the land are so important that most plants today still have these mycorrhizae, these fungus that live in them with a symbiotic relationship. And I'll talk about that. I'll talk about that later too. Mycorrhizae existed back then. They exist today. The biggest problem, of course, moving into the drier areas of the continent is drying out, right? So leaves begin to develop things like waterproof coatings. Our red mangroves that grow in Florida get waterproof coatings. If you touch one, you can feel the waxiness. The best thing that was going on for at least the living organisms and other plants is as the plants began to move out first, they started making new opportunities for everything else. There's this thing in ecology that we call certain species of plants are known as pioneer plants. Pioneer plants are ones that are really good at living at sort of harsh environments, and then many times what they do is they'll change the environment. They don't do it because they're thinking about further plants in the future, but because of the chemistry of that plant, the environment gets changed and other plants can now move in. And when plants move in, then animals can move in, and then when animals move in, pollinators can move in, and blah, blah, you get the idea. Right, so plants are creating opportunities for organisms to move around. This is a picture of mycorrhizae. This is the cell. I'm sorry. This is the root of the plant, and all of this around there is fungus called mycorrhizae. Not a damaging fungus, a beneficial fungus. Okay. I asked you guys in class the other day how many extinctions we had, and you guys told me I think 2. 5 are recognized. We may be in the midst of a 6th, but the 5 we have one at about 440 million years ago. Okay, and then another one about 360 million years ago, and then this one, which I am going to talk about, that was a big one. That's the Permian, 250 million years ago. What I got there, 3, 4. This one happened during the early part of the dinosaurs. Doesn't mean the dinosaurs went extinct, but they did go extinct here. Now, I'm only going to talk about these 2, this one here and this one here. I'm going to talk about the Permian and the Cretaceous, which I don't want to spell Cretaceous out, but it's a long word. Anyway, well, there it is right there. The Permian and the Cretaceous tend to be the most notable. The Cretaceous, not necessarily because it's the biggest, because the one back in the 444 million years ago, it was actually bigger, but because it had a tremendous effect on what happened on Earth. Remember, the dinosaurs were doing everything back then. I got 2 minutes. Let me get into the Permian extinction and see if we can talk about that. So 250 million years ago. So I'm talking to you. We've got this rapid diversification of life. Everything's going along fine for 300 million years. What happened in the Permian that allowed this change in the environment that eventually allowed us the dinosaurs? Here's what happened during the Permian. Here's the battle report. 90% of all marine species went extinguished. That includes the fish and all the other types of marine species. A third of the insects were gone. Happened in 5 million years. So the Cambrian explosion was 25 to 100. This was significantly less. And again, in geology, that's what we use these extinctions in some cases to draw boundaries between the Paleozoic right here and the dinosaur era. Always remember the dinosaurs are the Mesozoic. What caused this? Well, one of the things that we think caused was the formation of land. I know that seems odd, but the formation of land into a particular arrangement known as Pangea. Everybody's, I'm sure, aware by this point that the Earth is made of these crusts that float around on this semi-liquid surface, and those crusts bounce into each other. And so what that has done is that's changed the way land is arranged on Earth from the beginning to the way it is today. All right, so I'm at my time right there. I'm going to stop the recording.